Good morning, everyone. Today we will be seeing the haplodiplontic. Life cycle pattern. Now, in this, right? In this, both haploid and diploid. In this, everybody keep on your videos. Whose video will not be on, I will mark this your name and I will give it to your class teacher. Everybody mark, put on your videos. Aarti, Bhavya, Harshita, Ayush, Madhvi, Upak, Ayushi, Anjali. Still some videos are not on. Raj and Anjali. Can I write your names now? Ayushi Chaudhary. I think she is not there. Ayushi and Raj Nandini, Anjali. I cannot see you, Ayushi. Raj Nandini. Okay, your names I am going to write. Bhavya, again your video is off. Harshita, Kanchan, Jia. Okay. In this, both haploid, in this, both haploid and diploid phases. Bhavya is not here, Anjali. You all will be marked absent now. Ayushi Chaudhary, Anjali, and Jia. Okay.
Miss, voice is not clear.
In this, both haploid and diploid phases are. Others can hear me nicely.
Yes, miss.
Then it means some network problem will be there who is not able to hear me properly. In this, both haploid and diploid phases are multicellular and often free living. Often free living. Means haploid phase, that is the gaptophytic, and diploid phase was sporophytic. These phases will be free living, not dependent on one another.
Yes, please repeat it.
In this, both haploid and diploid phases are multicellular and often free living, but they differ in dominant phases. Which will be the dominant phase? The phase in which the plant is doing photosynthesis. That will be the dominant phase. So in both, the dominant phases can be different. Next para. In bryophytes, dominant photosynthetic thalloid plant. In bryophytes, dominant photosynthetic thalloid plant body represents haploid gametophytic phase.
Miss, please repeat once again.
In bryophytes, dominant photosynthetic thalloid plant body represents haploid gametophytic phase. Sporophyte in bracket capsule. In bryophyte, the sporophyte is what? It is a capsule. So sporophyte in bracket capsule is short-lived, multicellular, independent or partially dependent, or partially dependent on gametophyte. Next para. In pteridophyte, diploid sporophytic in pteridophytes, diploid sporophytic photosynthetic phase. In pteridophytes, diploid sporophytic photosynthetic phase is dominant and independent. Gametophyte in bracket Prothallus. Gametophyte in bracket Prothallus. In gametophyte in bracket Prothallus is haploid, short-lived, and independent.
Miss, what you said after Prothallus?
Is haploid, short-lived, and independent. Example, algae. Such as Ectocarpus, Polysaccharide. Yes. These are all algae. So all these bryophytes, pteridophytes, and the algae like Ectocarpus, Polysaccharide, and Kelps, they will have the haplodiplontic phases of life cycle. In pteridophyte, the dominant phase will be the sporophyte, it will be diploid, and in bryophyte, the dominant phase will be the gametophyte. So life cycle will be drawing. life cycle patterns are very important. Anyone definitely likes to ask. Now for the life cycle, graphical representation of life cycle of angiosperm. You will be studying from your book itself, page 3.35. Open your book, page 3.35. I will be explaining in the book itself. I'm not going to draw the diagram here. So all of you look into your book and I'll explain it. All of you open your book, page 3.35. Have you opened it? Again, many videos are of Anjali, Anshika, Anush, Bhavya. Bhavya is continuously not started, so it means she's not attending the class. Jia also continuously videos of Manglesh, continuously videos of Nidhi. So you are going to be marked absent every day when your videos are off. Anushka also not video is not on. It means you are playing your mobile and just absent from the class. Okay, so we'll be seeing we'll be understanding the life cycle from this diagram, page 3.35. So first of all, we'll be starting from the sporophyte. Look at the sporophyte. Sporophyte is the dominant phase here because this plant is going to do the photosynthesis and it is going to produce the flower. So this is the dominant phase and it will be diploid phase, 2N. All the cells will be diploid here. And it will be doing the photosynthesis. It will produce the flowers, and flowers will have male reproductive organ and female reproductive organ. So we can see here the ovary and the stamen. So inside the ovary, if you see the arrow, there will be the megaspore mother cell. So this I will be explaining here. This part only. Inside the section, pollen chambers. These are the pollen chambers. The anther sac. This is the anther sac. These are anther sacs. There will be 4 chambers in 2 anther sacs. So 4 chambers I have drawn here in the cross section. And each chamber will have a hollow area, tissue filled area. There is called as pollen chambers. Inside this pollen chamber, there will be the microspore mother cell. Are you able to hear me? Earlier, this is filled with the tissue cells. This is filled with cells, parenchyma cells. One of the parenchyma cells will become Microspore mother cell. Any part of the mother cell will become microspore mother cell, and the nucleus of this microspore mother cell are not splitting all, are making only this one, which is becoming microspore mother cell. And this microspore mother cell will undergo meiotic division and it will produce the pollen. Not here, actually. It's produced in the form of microspore. They get separated from one another. They produce the pollen grain. Okay. So this is how the pollen grains produce the male gametophyte. This pollen grain is called as male gametophyte. Is it clear? Pollen grain is the male gametophyte, and it is going to produce the male gametes, that is the male nuclei, which we have studied in class 9. Similarly, pollination is the only one. So in the beginning, the ovule was like this. Was filled with parenchyma cells. And this part of the ovule, this part of the ovule is called as microhydral end. And this is called as serosal end. So this is filled with parenchyma tissue. There will be the outer layer epidermis, and inner layer hypodermis. This will be made by cells. These layers will be made by cells, epidermal cell and hypodermal cell. So, one of the hypodermal,
Voice problem.
yes,
Voice problem is there. Something is
whether your network will be, all are having the voice problem, all are having the voice problem. Just raise your hand or unmute and tell.
No, miss.
Teachers are teaching from here only and all.
No, miss.
If anyone is having the voice problem, please check your network quality, etc. Okay. So this ovule is having the outer layer epidermis, inner layer hypodermis. This is the ovule before the formation of egg. So it is the primary ovule, we can say. So one of the cell, these are the cells here in the hypodermis. So one of the cell, this cell, towards the microhydral end, it will start functioning as megaspore mother cell. The cell of the hypodermal layer towards the microvillus end will start functioning as megaspore mother cell. Then what will happen? Will develop in segments. This megaspore mother cell will become large. It will become large and fill the whole space. The megaspore mother cell of the hypodermal layer will become enlarged and will fill the whole space here. And this will be called as embryo sac. From which the embryo sac is formed? By the enlargement of megaspore mother cell. And this megaspore mother cell is what? It is the cell of hypodermis towards the microvillus end. So, and the nucleus of this megaspore mother cell will undergo meiotic division. Because all the cells here are diploid, so the cell of megaspore mother cell will undergo meiotic division and it will produce, first of all, cells, then 4 cells, then 8 cells. It will become 8 nuclei. From one nucleus, first division of nucleus becomes 2 nuclei. Second division of both the nuclei produces 4 nuclei, and the third division of all the 4 nuclei will produce 8 nuclei. Okay? Then these nuclei will arrange them. They will move towards this region. 3 towards here, 3 towards microvillus end, 3 towards stellate end, in the center. Are you getting the concept? What is happening here? The megaspore mother cell. Which cell it is? The cell of hypodermis towards the microvillus end of the ovule. So this cell will become enlarged and its nucleus will undergo 3 repeated divisions. So first of all, one more thing is happening here. Before it becomes enlarged, it will undergo meiotic division. When it will undergo meiotic division, it will produce 4 daughter cells in line. 3 of them will degenerate and 1 will survive. 3 towards the microvillus end will degenerate and 1 will survive. This one which will be surviving, its nucleus will undergo mitotic division and cell will enlarge. Nucleus will undergo mitotic division. 8 nuclei will be formed. 3 towards stellate end, 3 towards microvillus end, N2 is the center. Again, I am repeating it. What is happening? The hypodermal cell towards the micropylar region, it will undergo meiotic division. It will be the megaspore mother cell. After the meiotic division, 4 daughter cells are produced. The 3 daughter cells towards the micropylar end will degenerate, disintegrate, and the 4th one will be surviving. This 4th one will become enlarged and form the embryo sac. And its nucleus will undergo 3 repeated divisions. First division produces 2, second division produces 4, and third division produces 8 nuclei. And these 8 nuclei will arrange in different regions of this megaspore mother cell. So, 3 will arrange towards the chilazal end, 3 will arrange towards the micropylar end, and 3 will arrange in the center. So these 3, now after this arrangement, the cell wall will be formed. So they will form the cell wall. And they will become 3 cells here, 3 cells here, and 1 cell here. So it will become 7-celled embryo sac. C towards another end will be called as antipolar cells. 3 towards the micropylar end, 1 will become end cell, and the remaining 2 will become synergies. And this will become polar bodies or secondary nucleus. This will become polar bodies and or secondary nucleus. So this is a mature ovule. By this time, the ovule will be mature, egg will be formed. And after that, what will happen? Next step in the life cycle, you see. So megaspore mother cell will form the 7-celled stage. You can see here. And the anther is producing the microspore, that is a pollen grain, or this pollen grain will be called as male gametophyte. And it will start germinating when it falls on the stigma, and it carries the male nuclei towards the micropylar end of the female gametophyte. Female gametophyte will be embryo sac. Embryo sac will be the female gametophyte, and pollen grain will be the male gametophyte. So when the pollen tube reaches the micropyle, it will dissolve it will release the enzymes. The pollen tube will release the enzymes, and the enzymes will dissolve the cell membrane here of the embryo sac. Then the nuclei will enter into the ovule, into the embryo sac through synergy. Synergic's role is what? It gives passage to the male nuclei to enter into the embryo sac. So through synergics, the male nuclei will enter into the embryo sac. One of the nuclei will fuse with secondary nucleus, and one of the nuclei will fuse with egg. And so there will be double fertilization. Then this egg will develop into embryo, and secondary nucleus will develop into the endosperm, and then embryo will develop. It will get nutrition from the endosperm, and when the seed is grown, it germinates into new plant. So this is how the cycle is completing. So this cycle you have to learn from your book. Only graphical representation is required. Only you have to learn the drawing, not to explain it. Only drawing and labeling is required. So with this, we are completing our this lesson. Now, all of you will try to solve the exercises given at the back of the lesson. It will give you revision also, and it will make you preparation also. So try to solve all the short answered questions, multiple choice questions, etc., definitions, etc., so that your lesson can be prepared.
Miss in rough copy, no? Or in
The book itself, you can go on writing the answers, put the tick mark, one word answer. In the book itself, you go on just for your revision. No need to write in any copy. If you want to write, you can write. But I'm not telling you to write because it is going to increase your work. Because already you have lot of work. So I will not tell you to go on writing all the answers. But if you will try to solve all the questions, you will be able to prepare your lesson. Now next lesson we are going to start. I'm not going to start animal kingdom because we might have got bored of studying only classification. So we'll just switch over to next lesson. That is the morphology of flowering plant. Animal tissue, animal kingdom will be studying later. So animal kingdom, we are not going to study now. We'll be starting the second unit, that is structural organization in animals and plants. And in this, we'll be starting the first lesson, morphology of flowering plants. So today I will not start this chapter. All of you will draw this life cycle in your copy. Okay? This life cycle should be there in your copy. And start preparing for your exams. Your exams, I think all of you might have got the notification that you are going to have your exams. I think from 26. Not got. You got the diary, no? In the diary it is written from 26. Not got the diary also. So you will be having your exams either by the end of this month or in the beginning of next month. Soon you will be getting the notification. So start preparing for your exams. So now I'm going to end the class. Okay. Take care and goodbye all of you.